All right, so let's talk about how fast a population of bacteria can divide. So bacteria as a population can grow extremely quickly, as you may already know. And this is because they can go through this replication cycle, binary fission, just really fast. And we see here E. coli has the shortest known replication periods, so it can divide approximately every 20 minutes as if everything is entirely perfect. So every 20 minutes. And that's, again, only if conditions are great. Not even if they're good. They have to be ideal. And there might be some other bacteria that could divide faster, but we haven't found them yet. It's definitely possible. There are also some bacteria that divide extremely slowly. I once worked with a bacterium that when it was in a certain stage, it would divide once every 2 weeks or so. So that's a really long doubling time. And what do I mean by doubling time? It literally means you get twice as many cells as you had initially. So one doubling time from here to here, that's one doubling time, then here to here. You're doubling the size of your population every single time. And this is what is called exponential growth, because you can measure this through a log. You could chart this on a log scale. So your growth would go up on a log scale. And you could determine how many bacteria are in your sample by using the number of original cells times 2 to the number of generations. So let's write that out quickly in a way that will be helpful. So this is the number of original cells. So we have one cell originally, and we're going to multiply it by the 2 and raise to the number of generations. How many generations do we have? We have one generation first, and so that would equal 2 cells, and that's what we see here. If we do it for the second generation, we started out with 1 cell. We multiply it by 2 to the number of generations, and so the number of generations is 2. So that would be 4. And so we see 4 cells here. That's correct. So you should know this formula. You don't need to write it like this, but you should be able to use this formula to calculate the number of cells in a population. Normally, we're not going to start out with a single cell, but it's fine to start out with that for our examples here. Other things that are really important for bacteria related to their fast replication time is that they tend to have smaller genomes than eukaryotes, so it takes them less time to copy their DNA. They don't have large complex organelles like many eukaryotes do, so they don't have to replicate those either. And in addition, they don't have to export their mRNA from the nucleus and then have translation occur. And we're going to talk about this in the future, but I just wanted to mention it here since it relates to what we're talking about. So for bacteria, they can have transcription and translation happening at the same time. So we have bacteria here, we have our DNA, and of course, we know it's circular. I'm not drawing it that way just because I want to save time. And we could have our mRNA being made here, and this might be a long piece of mRNA, and our ribosome could be reading the mRNA and making the protein, even as it is being transcribed. So there are a number of factors that allow bacteria to replicate really quickly. These are some of them. Again, the fact that transcription and translation can occur simultaneously, the fact that their genomes are smaller. I'll say they're kind of small. And the fact that, and we'll talk about this in the future as well, the fact that some of their genes are on operons. And so we could have more than one gene on each piece of mRNA. So more than one gene on one mRNA. So just as I drew here, I drew one continuous piece of mRNA, but maybe it has 3 genes on it, and those all get transcribed together and they all get translated together. So I think that makes it pretty clear why bacteria can replicate so quickly, so why there's so little time in between their generations. And as we know, bacteria don't need to have any kind of development. They're single cellular organisms. They don't specialize. They don't have any cells that specialize. So that's another reason why it takes eukaryotic organisms and multicellular organisms longer to have generation times, because their cells need to specialize. They have special compartments and specialized proteins. Their cells need to work together. And in bacteria, they don't.